Hi all, we're going to start where we left off on theme 5, which is the book's core theme that states that evolution accounts for all of the unity and diversity in living things. So we're going to start out with talking about how life is organized, how scientists want to put living things into categories to help organize their similarities and differences. So we'll talk a little bit about taxonomy, and then we're going to talk about evolution and how Charles Darwin really helped solidify a mechanism to help explain evolution. I do want you to take just a second and look at this cool picture. It's an artist's rendition of a tree of life, actually from the pedigree of man. So if you look down at the roots of the tree, those are the common ancestors, those were the first animals. And then as you move up the tree, you see animals get a little bit more, first of all, diverse, and secondly, complicated, bigger, intricate. And you go up to the next layer and you see fish, and then finally go up to where you see mammals and humans. So we're going to be able to look at trees similar to this, not drawn like an actual tree, and that will allow us to interpret evolutionary relationships based on a tree like this. But let's start first with classification. Scientists like to classify things. They like to put things in groups so that they can better understand that they have certain similarities because they're in a particular group, or maybe that they have differences. So one of the ways is to use the science of classification, which is called taxonomy, to help us figure out what organisms are more closely related to others. Now remember, we talked about if we looked at all the cells in the world, they shared some characteristics: cell membrane, cytoplasm, ribosomes, DNA, and that DNA coded for proteins. We could take those cells and divide them into two categories: the prokaryotes, which were simple, not a lot of structural organization, and the eukaryotes, which had a lot more structural organization. Now, within those categories, we can even further classify and specify similarities between organisms under those big umbrellas. And we use the taxonomic system to help us do that. So the taxonomic system uses the, let me get a pen that you can see. It's called domain, kingdom, phylum, class, order, family, genus, species. And each of these categories is inclusive of certain organisms, but as you go to the next level of categories, this one is less inclusive. It doesn't include as many. So let's start here. This is the domain we call eukarya. It includes all eukaryotic cells. So we know that any cell that's in this category has a nucleus, has a mitochondria, right? Maybe has an ER and a Golgi. But you can see here when we look down, even though they all have all those things, they're very diverse. We have bears and a wolf and a zebra and a frog and a bird and a turtle and a starfish and a sponge and a flower and a mushroom and a single-celled organism. All of these have that nucleus and those subcellular organelles that put them into this category. So if we want to get a little bit more specific, we can move into the next category, which is the kingdom. So in the domain eukarya, there are 4 kingdoms: the animal kingdom, the plant kingdom, the fungus kingdom, and then the last one, sometimes not really called a kingdom, we'll talk about it later. It's a group called the protists. All right, so if we look at this picture, we are going to look at just the organisms that are animals from this domain. So notice we're going to remove this single-celled organism. We're going to remove this daffodil, it's a plant. We're going to remove this fungus. And everything that's left here is an animal. And yes, a starfish and a sponge are both animals. Now we want to get more specific, and we're going to move to the category called a phylum. And we only want to look at organisms that have backbones. So again, if we go down here, sponge doesn't have a backbone, starfish doesn't have a backbone, butterfly doesn't have a backbone. So we're going to look at just the ones with a backbone. Now we want to get even more specific. So we're going to look at the class, and in this case, the class is called Mammalia. Only going to look at mammals. They have hair, they give live birth, they have mammary glands. Well, birds, toads, turtles, fish don't do that. And so we're going to just limit us to the ones that do. Now, of these ones, we only want to look at the ones that are carnivores. Well, zebra is an herbivore. A dolphin is an herbivore. So in the order, we're only going to look at carnivores. Then we're going to move to a family of organisms that are very similar to bear-type organisms. And then specifically, these bears, black, grizzly, and polar, are in this genus. And then we get to the most specific category, which is the species. And remember, we told you that a species are organisms that are similar in structure, function, DNA, things like that. And when they mate, they can have viable, fertile offspring. We typically refer to an organism by their genus, species, name combination. So you can see here for this black bear, the genus is Ursus. The species is Americanus. So his name is Ursus americanus. And these are names that are agreed upon across the world, so not just in the United States. So this is taxonomy. So how are you going to remember this? You don't necessarily, I'm never going to ask you to write these in order, but I might say, are two organisms found in the same class more closely or less closely related than two organisms in the same kingdom? Right? And you would say they're more closely related because look at all these extra ones in this kingdom. So the way to remember this is, did King Philip come over for good spaghetti? And that will help you remember the order that these flow in. All right, let's look at this. Species that are in the same blank are more closely related than species that are only in the same blank. So we need to find something that is a smaller category compared to a bigger category. So is phylum smaller than class? No. Is kingdom smaller than phylum? No. Is family smaller than genus? No. Is class smaller than order? No. But family is smaller than order. So if you're in the same family, you're going to be more closely related than those that are in the same order. Does that make sense? All right. Now, we use the similarities among organisms to help group them. And originally, people used anatomical or maybe developmental type similarities to group organisms together. But as we've learned more about molecular biology and DNA and proteins, and we've learned that we have a sequence of DNA and a sequence of amino acids in our proteins, we find that we can use that information almost like a measuring tape to tell us when there were changes that caused organisms to diverge and be different from each other. And we can represent those changes in a tree like this. This is called an evolutionary tree or a phylogenetic tree that represents those three domains that are present in taxonomy. So their first domain is called domain bacteria. Another domain is called domain archaea. And the last domain was the one we just spoke about, which is domain eukarya. Domain bacteria and archaea are all prokaryotic cells. Domain eukarya contain the eukaryotic cells. And you can see here, this represents those four kingdoms or groups, right? The animal kingdom, the fungus kingdom, the plant kingdom, and then this other grouping called the protists that are eukaryotes that aren't animals, fungi, or plants. So when we look at a tree like this, it's very similar to that tree I showed you in the previous slide. Down here at the base of the tree, where the roots would be or where it touches the ground, this is a common ancestor. This goes back in time. And as we move up, that's evolutionary time right there. And then right here at this branching, there were enough changes in the DNA that accumulated over time that some of the organisms differed, and so they branched off from each other. So every time you see a branching like that, that's what happens. And if you draw the tree appropriately with using data to draw it, the length of these branches indicate time. So there's a reason why this branch is this long before there was this purple domain archaea broke off. And then this branch is very long before we actually broke off into the eukarya. So this is called the tree of life. It represents a common ancestor that branches off into 3 main branches. Domain bacteria off on its own to one side. Then the other branch off to the other side has first archaea and then eukarya off of it. Now, one of the things that we'll notice when we look at living things is that despite the fact that they all are made of cells with a membrane, cytoplasm, DNA, central dogma, all that stuff, there's also this amazing amount of diversity. So it's kind of an interesting and abstract thought to think that they can be so the same but so different at the same time. This is a picture of Charles Darwin, who's considered the father of evolution. Evolution helps us to explain how those living things can be so similar yet so different at the same time. Our definition of evolution is that organisms that are alive today are descendants of organisms from previous generations. What Charles Darwin did was he described a mechanism to explain how evolution works. Evolution was not a new idea, but nobody really could figure out how it works. And one of the amazing things that you have to really think about was that Darwin was figuring this out before anybody knew that DNA was the genetic material, before anybody knew the central dogma, meaning that DNA directed proteins to do things, before anyone knew that proteins were really responsible for all of our traits and functions, even before we knew how cell division worked. So the fact that he figured this all out kind of in an abstract way without knowing a lot of the facts that we now know that support his ideas was pretty amazing. So a little bit about Charles Darwin. His dad was a surgeon, and in those days you did what your dad did. So he went to university to become a surgeon, just like his dad, and he really hated it because this was before there was anesthesia, and so it was not a fun process to be a surgeon. And so he was a little bit lost and didn't know what he wanted to do. He was 22. He was at university. He was definitely a curious guy, liked science, just didn't want to be a surgeon. And one of his professors came to him and said, a bunch of us are going to go on a voyage to map the New World. So basically they were going to map the coast of South America. And do you want to come along since you kind of don't know what you want to do with your life? Maybe this can help. And so he went on this 5-year voyage to map the coast of South America. And they went across the Atlantic Ocean, stopping at islands along the way. They started at the northern coast of South America, and they just basically stopped all along around both sides of South America. They actually went to New Zealand and Australia, and then they came back to England. So it took them 5 years. And his role was he was considered basically the ship's naturalist. So when he got off the boat every time, he would just walk around and take note of the organisms and living things and environment that he saw, and he took really good notes. And so he would say, okay, I see birds, and they're eating insects, and I dug in the ground and I found fossils of seashells, and here are some weird looking birds that have funny looking feet that are eating worms, right? So he was just paying attention to what they were doing and what they looked like and taking notes about it. And he did this every single place they landed for 5 years. And from that trip, he made some observations that we're going to talk about in just a second that led him to write the book called The Origin of Species, which basically presents a mechanism to explain the process of evolution. So that mechanism was called natural selection. And basically what he was suggesting was that when organisms look different from each other, that gives them some sort of either advantage or disadvantage based on the environment that they live in, and that the organisms who are better suited for their environment are the ones that are more likely to survive and pass their their traits onto the next generation. And over time, organisms of a particular species might change drastically because of what traits are being selected for. All right, so what did he really observe? So Darwin made these observations everywhere he landed, islands, mainlands, inlands, north, south, right? Everywhere he went, he saw these things. First thing he saw was when he looked at a population. So this is important. Natural selection is something that happens at the level of a population. When he looked at the population of, let's just say, birds on a given place, he found that among the birds there, so let's just say he was looking at finches, they were not all identical. There were individual variations within the population. So just think about humans, right? We don't all look alike. We have different skin color, hair color, eye color, height, weight, whatever, right? We're all a little bit different. And he noticed that no matter where he went, and really no matter what organism he looked at, that there were individual variations. He also noticed that in general, organisms made way more offspring than they needed to. So there was this overproduction of offspring. In an evolutionary sense, to be successful, you basically just have to reproduce to replace yourself, right? Well, organisms make way more kids than they need to replace themselves. Think about a litter of kittens, right? Maybe in a female cat's life, she might have 8 to 10 litters over her lifetime and 5 to 7 kittens in each litter. She only needs one to replace herself, but she might have that many cats. So what he started to notice this over and over and over again. So what he came up with to explain how evolution might be supported by this is he thought, if everybody in the population is a little bit different, maybe you have, let's think about a litter of kittens, maybe you have a runt and you have kind of 2 or 3 that are maybe normal sized, and then you maybe have a couple that are maybe overgrown, maybe a little bit on the bigger side, right? Those kittens in that litter are not going to have the same chance at being reproductively successful. Because in order to be reproductively successful, you have to find a mate. And in order to find a mate, you have to have energy. In order to have energy to go out and find a mate, you have to have eaten. And in order to have eaten, you have to have not been eaten yourself by a predator, right? So if you think about the kittens, maybe the runt just isn't fast enough or quick enough to get away from the predator and so can't get food. And maybe the big one gets food and is big but cannot. So if the runt gets eaten by the predator, that runt isn't going to be reproductively successful. If the big one is too slow to get away from the predator, they're not going to be reproductively successful. And so maybe only a few of them survive, and the ones that survive then are the ones that pass their traits to the next generation. So what happens is you get this accumulation of favorable traits over time. So let's look at an example. Here are a bunch of beetles, a population of beetles. They live on a background that's black. And you can see there's a little bit of variation in their color. Some are lighter, some are medium colored, some are darker. Now a bird eats these beetles. So a bird's not going to land and kind of do what we do at Walmart and look at the shelves and think, what kind of cereal do I want to eat this week, right? The bird's going to have to hover from above to say, oh, there's some lunch down there. And it's going to swoop down quickly, pick up a beetle and go. If it landed, it would scare the beetles, or it could get attacked by a predator. So it has to just be able to pick its food from far away. So obviously the ones it's going to see from far away are the ones that have contrast or difference from their background. So it's going to swoop down and eat the lighter colored ones. The lighter colored ones then at that point are not going to be able to survive to reproduce because they've been eaten. The ones that are left are capable of surviving to reproduce. So over time, what you would find is that the beetles in that particular environment would likely be more suited for their environment so that they didn't get eaten, because the darker ones are what survive. Now, this is completely dependent upon your background or your environment, right? If all of a sudden we change the color of the background under the beetles, like maybe you went from a blacktop driveway to a a cement driveway, all of a sudden a different beetle would stand out from the background and the birds would likely eat the other beetle, right? So it's nature doing the selection, in a sense. The environment, what the environment looks like and the conditions of the environment are what determine the traits that will be favorable for those particular organisms. And environments are always changing. So there is no perfect, dark beetles are that perfect beetle, right? There's no perfect trait because environments are in a constant state of flux. So let's answer this question. Which of these individuals is most likely to be successful? Remember, what did we talk about? Successful in an evolutionary sense. A male who mates with 20 females and fathers 1 offspring. An organism that lives 100 years and has 2 offspring, and both of them survive to reproduce. A reproductively sterile individual who never gets sick. An organism that dies after 5 days of life, but leaves 10 offspring, all of whom survive to reproduce. Or a female who mates with 20 males and produces 1 offspring. So think about that for a second. What do you think the right answer is? Probably you're debating between B and D, because both of those produce offsprings that reproduce, right? But which one is probably better? D. If you have 10 offspring that pass your traits on, that's being more evolutionarily successful. So Darwin made 2 main points. He talked about the idea of natural selection, the mechanism that drives evolution, and sometimes he referred to it as descent with modification. So how, you have descendants, but they're not quite like everybody in the original population, all right? And this helped to explain how you had to have a cell that had a membrane and DNA and cytoplasm and all that, but the way that cell's traits were could differ from population to population, from environment to environment, from organism to organism, and that's how you got all of the diversity. This is just an example of unity and diversity. So these are the bone structures of mammals, and you can see here that we have a 1 bone, so your humerus bone, 2 bone, kind of lots of bone, and then finger bone pattern of bones in our arm. Think about what we do with our arm. We write, we throw, we cook, right? We have fine dexterity with our fingers. We can do things. If you look at a cat has the same pattern of bones, 1 bone, 2 bone, lots of bone, finger bones. Cats can do things very differently than we can. They don't write or cook, but they can clean themselves, they can bat at a toy, they can climb a tree. If we look at a whale fin, the bones look different, but the pattern is the same. 1 bone, 2 bone, lots of bones, finger bones, and that allows them to more easily move their fins through the water for swimming. And in a bat, look how skinny the bat bones are. 1 bone, 2 bone, lots of bone, finger bones, because they need to be light because they have to fly. And they are there to kind of separate the wing structure so that they can take flight more easily. These are all from a common ancestor. Somewhere in the line that 1 bone, 2 bone, lots of bone, finger bones worked, right? And then as environments changed, some of those got thicker and shorter. Some of them got thinner and skinnier. Some of them were shaped a little bit different in the way they were positioned with each other, but the pattern stayed the same. Okay, so this is something called, well, let's ask you that. What does this represent? We saw this picture kind of earlier. It looked a little bit different. This would represent a tree of life, right? An evolutionary tree, right? And so let's ask ourselves what these things might be, because you're going to have to be able to recognize and at some point even create phylogenetic trees like this in your future. So these letters up here, A, B, and C, represent the 3 domains of life that we learned about. So what domain do you think is A? What domain do you think is B and C? Of these domains, which ones of them are prokaryotes? Which one is eukaryote? What does D mean? Where would you fit on this tree? So think about those questions. If you want, you can send your answers to me and I'll tell you whether you're right or wrong. And just know that at some point you might have to draw a tree like this. So be prepared to represent the 3 domains in this format. Okay, the last part of chapter 1 is the process of science using scientific inquiry. And this is just kind of a flow chart of how it works. So when you're going through a scientific experiment, it flows in this way. And if you follow these steps, even if you don't get a result, the result can be meaningful for whatever you are looking for. So the first thing is, most of the time it starts with making some sort of observation. You discover that something happens a particular way and you start questioning it, like, wonder why that happens this way, right? And so then you might go and you might research it a little bit, maybe Google search it, or ask your teacher, or talk to your friends about it, or ask your mom, right? And then you might come up with a prediction like, well, I bet if this is true, then this would happen, and you formulate what's called a hypothesis. From that, you would design an experiment to actually test your hypothesis. You would collect data, analyze that data, maybe go back to the people that first gave you information and said, look what I found, like, wonder how this fits in with all of it, and then you would draw conclusions. Now in science, that typically leads you back to the conclusion, say, okay, here's a new observation which leads you to a new question, and you kind of stay in this cycle for a while. Eventually you collect enough data that you have something solid, novel to publish. So before you can actually publish it as a novel idea, it goes through a very stringent peer review process, where basically your ideas get sent out to others who are in your field. Not necessarily, doesn't mean you're friends when it's peer reviewed. You don't get to choose who it is. And those people critique it, and they're pretty harsh. They say, wait, we need you to do statistical analysis, or we think you need to do this again with these control groups, or something like that. So you have to go back to the drawing board sometime to address their questions. And then eventually that group of peers will approve that you've done, sufficient to show these results are valid, and then you would publish it into the scientific journal world. And that's where we go when we want to find out information about things that scientists have done in the past. When you're doing experimentation, you have to make sure you're doing it in a controlled way. So you have to have a control group, where you're not going to change anything, and then you're going to compare that to an experimental group. You have to always consider, when you're going in, your variables. What are you changing? That's your independent variable. And then how are you going to measure the difference, right, from what you changed and what in your experimental group, what you didn't change in your control group, how you're going to measure it. The measuring is the dependent variable. One way to remember this is the independent variable starts with the letter I. That's what I change as the scientist. The dependent variable changes as a result. It depends on the independent variable. So make sure that you read and understand a little bit more on the scientific method. It's covered more thoroughly in lab, so it's both places. Make sure you have an understanding of the difference between inductive and deductive reasoning, and which one scientists typically use. What's the difference between qualitative and quantitative data? One thing I really do want to make sure you know the difference is between a theory and a hypothesis. So make sure that you understand the differences between those things. In lab, in particular, you'll talk specifically about the difference between theory and hypothesis. Feel free to shoot me an email if you want to say, okay, here's what I found. Here's the difference between inductive and deductive, and qualitative and quantitative, and theory and hypothesis. Am I right? Don't just email me and say, what is the difference? Figure it out. Go read it in your textbook, look it up online, and then write your answer to ask me if you're right, okay? This is just, eventually you're going to graph your data. So this is just another one of a thinking question for you. So when you graph your data, you're going to basically be graphing the independent variable and the dependent variable on your graph, so that you can say, here's what I changed. Maybe I changed the concentration of sugar in my solution, and then here's what I found as a result. As the concentration went up, the growth changed in this way of my organism. So you have to know how to put that into a graph format in lab. So looking at this, kind of maybe think about, where would you put the independent variable, what I changed, and then where would you put the dependent variable, and see if you could label that. And again, you can send me your answers to see if you're right. Okay, that's it for theme 5. Let me know if you have any questions.